Okay, are we ready, Lucy, from your side?
Yeah, ready over here.
Terrific. Okay. Right. Well, hello, everybody, and welcome to our session 3, third session of the day, shallow water processes and sediments. Let's just go over those rules again. It's 12 minutes for presenters. Please, presenters, let's stick to the 12 minutes. Otherwise, you will be severely cut off. Quite literally, your video stream will be cut. You will get a 2-minute warning. You will get a 2-minute warning from myself or from Lucy, whoever's introducing you. Then we'll be having 7 minutes, just 7 minutes of questions. Actually, it's quite a long time. So please, members of the audience, do ask your questions. Remember, these are the guys that are at the cutting edge of the research. They're doing the stuff that we all wish we could do. They've got the time, they've got the energy, and they've got the brains to do it. So please do ask them questions, give them pointers, give advice, sure. Right, okay, so without further ado, I'll ask the first of our presenters to step to the microphone. So it's a Haiwei. Okay, if you could do your screen sharing now, please. Okay. So Lucy and I'll turn our videos off. Okay, Haiwei, you have your 8 minutes. Sorry, your 12 minutes. Start when you're ready. Terrific.
Okay. Thank you, Stephen. And hello, everyone. I'm Haiwei and from University of Liverpool. Today, I will be talking about spatial self-organization of lacustrine carbonate microbial mounds with insights from our stratigraphic forward model, Stromatolite 3D. And this project is funded by Equinor. So in today's presentation, I will start from some background introductions to microbial life and spatial self-organization. The key questions are whether spatial self-organization is present in microbial life and why does it matter? Microbial life are one of the oldest fossils in Earth's history, and they also form important hydrocarbon reservoirs globally. Here's an example from Campos Basin offshore Brazil. We see microbial life on inner part of the platform are dormal and isolated, but others on outer part are highly self-organized with respect to the shoreline and apparently connected in club and blade forms. And this preferential orientation suggests a self-organization possibility, and it will be important for primary strata anisotropy. So understanding the fundamental processes for microbial life occurrence and distribution can provide useful implications for reconstructing paleo environment and predicting species heterogeneities. Microbial life are organosedimentary deposits that have accreted by benthic microbial communities, usually have some internal fabric laminated or clotted, and they can form by precipitation or green trapping on sticky microbial mass. The water chemistry controls the rate of precipitation. Abundant extracellular polymetric substance, EPS, produced by microbes facilitates green trapping under agitated conditions. Those are the important factors controlling microbial life growth. And spatial self-organization is a process in which coherent pattern emerges through interaction of system components. It is pervasive in sedimentary systems, but still poorly understood and documented in carbonate systems, especially for microbial carbonate, with a few exceptions. And a recent study of present-day algal mass on intertidal flats, functionally equivalent to microbially induced bed forms, show internal laminations and regular morphological patterns that are strikingly similar to microbial-like strata found in geological record. And this rich and runny patterns implies that self-organization was an important morphological process in times when biofilms were dominant. Here we use stratigraphic model to understand which processes are required to create those spatially ordered patterns and their implications. So, an introduction to our model, stromatolite is a small-scale, fully deterministic model, and it includes a hydrodynamic model with a dominant flow direction to represent conditions under strong wave or tidal influence environment. And this multiple water flows, the entering the model edge will follow this dominant direction, but also with local variations caused by wave refraction. And the flow velocity is defined as the flow density of each cell on the model surface. So we see flow velocity is higher when these flows are converging. And we assume that fine-grained or nocturnal sediments in suspension are entering the model at a user-specified constant rate, and their depositional rates depend on the surface gradient and flow velocity. And they also provide sediments to be trapped and bind into microbial light, but they can also inhibit microbial light growth if their concentration in the water column is too high and decrease the light level. So the rate of microbial light growth is controlled by the amount of sediment in suspension, highest when the suspended sediment just start to block light penetration. And also it's controlled by flow velocity, because trapping and binding favors a high energy agitated condition, but excessively turbulent water will be negative. So those are the model inputs. This is the initial surface. It's a random, smooth, random, irregular surface with relief up to ½ meter. And the initial water depth is 1 meter and it's rising slowly. The model formulation in those 4 experiments are different to represent increasingly complex spatial feedbacks from experiment 1 to 4. And experiment 1 has the simplest model formulation and the least spatial feedbacks. It shows a lot of classic small isolated mounds scattered across the whole surface, as we see in Shark Bay. And in experiment 2, we start to have a dominant flow direction, and the interaction between production and hydrodynamics produce fewer but larger mounds. And these elongated mounds are also observed in Shark Bay and referred to as longitudinal mounds. Experiment 3 couples the flow velocity, the hydrodynamics, and the suspended sediment deposition, and it develops those elevated ridges. Some of the ridges show Y-shaped junctions open to the incoming flow direction, like we see here in Shark Bay on the right. And it still contains some small untouched mounds. In the most complex experiment 4, the flow velocity, topography, production, and deposition of suspended sediments are all closely linked to each other. And this creates very regular and organized ridge and radial patterns indicative of self-organization. And small isolated mounds rarely occur, and the microbial lights are elongated and well connected in the dominant flow direction. And this is very important if those microbial lights are reservoirs. And quantitative evidence is required to identify and support the presence of self-organization. So here we calculate the topography correlation coefficient R to demonstrate the degree of dependence on initial conditions. So it is the percentage of the highest 20% of the points on initial surface that survive and therefore can be correlated to final surface height. In experiment 1 here, we see a lot of yellow blocks showing a strong correlation, and the R value here is just below 0.5. And in contrast, a more self-organized system will be less dependent on the initial surface. So the R value in experiment 4 is much lower. And the R metric also works if we select the highest 15%, 10%, or 5% of the points for calculation. And looking at these cross sections parallel to the flow direction, in experiment 1, the mounds are narrow and aggradational, implying a strong substrate control. And as the model is getting more complex and the spatial feedbacks become closer, the mound can prograde laterally, and we see these ridges start to develop, and the control of underlying substrate is much weaker. And in Shark Bay, the mound distribution varies from place to place depending on the substrate and hydrodynamic conditions. And from experiment 1 to 4, with increasingly complex spatial feedbacks, we produce strata pattern very similar to Shark Bay. From left to right, the substrate control becomes weaker, and there's an increasing possibility of spatial self-organization. There's also an increase of 3D connectivity from many small isolated geobodies to feel like larger, highly connected geobodies. So here we compare our modeled mound morphologies with observations from modern Shark Bay and also the Lower Cretaceous Coastal Formation in Northeast Brazil. And in both areas, the mound morphologies are diverse with different height-weight ratio. We replicate these morphologies in our model as a result of increasing vertical growth rate. And increasing the lateral growth rate facilitates sediment accretion interference and generates complex morphologies with these crowns. So the question is, what patterns will be produced if the substrate itself is highly organized? Here we run case 1B and 4B, which are equivalent to the experiment 1 and 4, except the difference in initial surface. And these elongated roots represent sand dunes, as we see in Shark Bay. And in both experiments, mound occurrence and distribution are strongly controlled by substrate. In cross-section, the mounds in 4B expand laterally, and we see the accretion is faster on the windward side. So the mounds are actually inclined towards the incoming flow direction. And the result here suggests even in a system with strong substrate control, the difference in modeled processes and their spatial feedback can still have an impact on morphology, among the morphology and faces heterogeneity. A new theory of spatial self-organization comes from the classic reaction diffusion theory by Alan Turing. And in his theory, local feedbacks occur between reaction components, but spatial information is also transmitted over a longer distance via diffusion. So formation of stable patterns requires the diffusion to be faster, requires the diffusion of inhibitor to be faster than activator. And in our model, microbial growth is clearly the activator, which occurs locally and is inhibited by the widespread suspended sediments over longer distance.
You have 2 minutes.
Okay, thanks. And in our model, like the topography and the flow velocity controls the microbial growth and suspended sediment deposition. And meanwhile, the topographic surface will also be modified after deposition. So the net feedback is scale dependent. Positive feedback dominates at short scale and negative feedback dominates at large scale. Okay, just to summarize, with the spatial self-organization can occur in microbial, and it requires interaction between topography, production, and hydrodynamics. It creates ordered, rich, and random patterns strikingly similar to observation in Shark Bay, with the implications for the connectivity. And quantitative evidence shows a decrease of dependence on initial surface in self-organizing systems. Substrate control, the substrate control and self-organizing feedback control are two end members, both of which control strata geometry. And spatial self-organization can be an important process and had an important influence on the stratigraphic record. So in the end, I want to thank my supervisor and our industrial partner in Equinor for sponsoring this project. And thank you. And any questions?
Thank you very much, Haiwei. Really, the questions are open. Audience, please do type your questions. Don't forget, please send the questions to everybody. If you send them to just SEDS Online, then the rest of us don't get to see it. So please send questions to everyone. And while we're waiting for those questions to start to come in, I got a very quick one. Some of these microbialites form not through nucleation and growth, but actually through an erosion process where an initial mat is eroded. So you basically erode into the side of a mat. How do you think that would change or evolve their morphologies over time using your observations that you've made in Shark Bay?
Well, actually, when I say like the hydrodynamics, it actually includes like deposition and actually entrainment. So you can basically like remove, like entrain a certain like fraction of the produced sediment and transport it into somewhere else. but what I found and what I've incorporated in this model is just the necessary processes. So in other words, those are the minimum elements required to create those patterns. And obviously, if you introduce some other processes like erosion, it's gonna like introduce some like complexities. And I also have like a routing, like I've described, like entrain some sediments and transport it to somewhere else. And it definitely controls the strata like heterogeneity. It basically increase the heterogeneity and decrease the connectivity. And from this project, especially the observations from the subsurface campus basin, there are lots of like branches related to this kind of erosion and transport process. But what I like to emphasize here is the minimum components. It's arguably like what happens if you include erosion, but the result here suggests like actually spatial self-organization can occur in this system without erosion. And so if you introduce erosion, it's gonna be, the strata geometry is gonna be different. Yeah. So.
Fantastic detailed answer. Thank you. Maybe we can have a chat at some other point. Let me go through the many questions that are coming in. You've obviously got people thinking. Volker Varenkamp, great talk. He wants to know, is the program available?
Well, unfortunately, it's not available online yet, but we aim to make this available to the public. Yeah. So currently, it's coded on MATLAB, and hopefully, like we can provide available on on some website here to everyone. So.
Thank you. A question from Alex Brazier. Alex Brazier is asking, thanks for the talk. Do you consider that the findings are specific to stromatolites like those of Shark Bay, formed by agglutination, i.e. sticking, trapping, and binding, or rather than chemical precipitation?
Oh, that's an interesting question. And well, I have to say, when I like developed this model, I'm slightly biased to the binding and trapping processes. And actually in the model, the growth rate is, I have like two elements of the microbial like growth. So like the precipitation growth rate is just a constant, and the trapping and binding process, which is controlled by the hydrodynamic and other processes. And what I found is that like in a stratigraphic formal model, if you want to model precipitation, you need to model very like detailed chemical processes, and it's not really realistic. I mean, in terms of the computational power. So, and that because the precipitation rate in the model is just a constant rate. So then when I present these results, when I said growth rate, it mainly refers to like it mainly refers to this growth rate associated with trapping and bonding processes. Yeah.
Terrific. Volker has another question very quickly. On slide 12, why do we create coalescing shapes that will break or impede flow?
Okay, let me just go back to slide 12. Well, I think like in this case, we see these mounds, they are elongated, like like parallel to the dominant flow direction. So connectivity is definitely improved in this direction. But if you like taking a cross section, like along a section perpendicular to the dominant flow direction, and it will still be kind of isolated. So I think that's the point. Like in this experiment, we see very strong like anisotropy with respect to the dominant flow direction.
Okay, we've probably got time for one very quick last question. We've got a lot more questions that are going to be unanswered, unfortunately, but thanks for the talk. This is from Cyprian, sorry, from Cyprian. Thanks for the talk. What are the simulation time steps and the carbonate production rates that you used?
Oh, okay. And it should be here, I think, the input. can't find it. All right, so I think the 10th step is basically like 1 year per so it's 1 year per 10th step. And the production rate, I think I also use the observation from Shark Bay. So it's between like 2 millimeter to 6 millimeter per year. Yeah, something like this. Yeah.
Great. I've just been told I've got time for one more question. Alana, hi Haiwei, nice talk. To further some of the questions on how widely the results are applicable, is this very specific to Shark Bay, and do you consider water chemistry?
Okay, I think this question is relevant to the previous question. I think like Alex asked, and well, I think like I said, it's because actually when I read it, it was actually like, because this project is funded by Equinor, so this model is aimed to understand the processes in the Campos Basin, and I think in this case, both the green trapping and chemical precipitation are important processes there. And it's just sometimes like it's a shame because precipitation is a chemical process, and stratigraphic form model usually focus on like physical process. So it's a bit difficult to combine these two. Yeah. So
that's great. Thanks very much, Haiwei. We still had outstanding questions from Jean, Dan, and Ismail. Guys, please do turn up at the discussion section after this session, and we can ask Haiwei those questions then. Have a good discussion about this and the other topics. So I'll hand you over to Lucy.
Yeah, absolutely. Thank you so much, Haiwei. That was brilliant. So we'll move straight into a talk from Daniel Gonzalez. Presume you're somewhere there in the audience. So he's going to be sharing with us on climatic implications of oncolite-rich beds in the Lower Permian Zottachkopf Formation in the Carnic Alps of Austria. So take it away. You've got 12 minutes.
Okay, thank you, Lucy. There we go. Okay, so thank you everybody for tuning in today for my presentation. Today I'm going to be presenting one of the chapters of my PhD thesis, which is about the importance of oncoites in the deglaciation that took place during the early Permian. And just a little bit of background here. So during the Mississippian and Pennsylvanian, we had different orogenies that built this mountain range kind of through the equator. You can think of the Avalonian, of course, the Hercynian orogenies. And most authors believe that the increased silicated weathering as a result of these orogenies facilitated the beginning of this late Paleozoic ice age, or LPIA, because it draw down a bunch of carbon out of the atmosphere. So the glaciation started somewhere in the Bashkirian. What happened later during the Astelian is that the glaciation reached its peak. And this peak coincided with a global sea level drop that is found really in sections throughout the world, in the US, China, here in Canada. And it also coincided with the lowest CO2 concentrations that have been reconstructed by Isabel Montañez and others. What happened later, the Astelian submarine boundary here, is the CO2 concentrations went up again, according to this reconstruction, this fairly narrow peak, and then it went down again, and then it went up on a steply fashion through the Erzincan and Congorian. So what we believe is that oncoites at this time may have played a more important role in the CO2 concentrations in the atmosphere than previously thought. So in order for us to look into this issue, we traveled to the Carnic Alps, which are located in the south of Austria. This is a classic carbonate location where many authors have worked. And the reason why we picked this location is because it records the icehouse to greenhouse transition. So there's two formations in this study area that are important. One is the Zweikoffel formation, which will be below the Zottachkopf. And the Zweikoffel formation is a mixed clastic carbonate succession, and it's cyclic. And the fact that it's cyclic, it means that it was deposited during the glaciation, during the LPIA. And we know this because glaciers glacial epochs have cycles of waxing and waning of glaciers. So when you have these cyclothems, these glaciostatic cycles, you know that it was deposited during a glacial epoch. Above the Zottachkopf Formation, we have the Zottachkopf, which is what we have here. And this formation is almost exclusively composed of limestone, some dolostones, and some clastics, but it's non-cyclic. So the Zottachkopf records, in fact, the deglaciation itself and then the post-greenhouse world. Oh, there we go. So this is the Zottachkopf section, the one that we measured in that photo that I just showed you here. And there's a few elements that are key for the purpose of this project. The first one is the sequence stratigraphy. So the sequence boundary that we first observed was at the bottom of the section. And this sequence boundary separates a lower clastic interval with some mixed clastic carbonate units, some karstic dissolution, festoon structures, and other kind of elements that would indicate a fairly proximal setting, above which we have open marine, very clean limestone. So we place the sequence boundary here, and then above the sequence boundary, all the way up to the top of the formation, there's a shallowing upward trend, or rather from the MFS. And this MFS is related to the transgression that was formed during the end of the late Paleozoic Ice Age. So it's a shallowing upward trend. And what is also relevant in this formation is the presence of oncoids throughout the formation, which is very important because what this is telling us is that oncoids at the time were present across the platform or across the ramp, not necessarily only in a lagoon or some kind of protected area. They were actually present throughout the formation or throughout the ramp. So these oncoids have been studied a little bit before by Flugel. Forky in 1995, he came up with a oncoid classification for the entire Lower Permian succession of this study area. Not necessarily only the Zottachkopf Formation, but this classification works for what I'm seeing. So I didn't want to reinvent the wheel, and I'm using the same classification for my project. So the first oncoid here in photos 1 and 2, these are mostly composed, or the cortices of these oncoids are mostly composed of Gyrvanilla, which is a photosynthetic or cyanobacteria. Photos 3, 4, and 5 here, these are oncoids that are composed of Gyrvanilla and Archaeolithophyllum, which is a red algae. Of course, these cortices are more complex than just these two organisms, but these are the main ones. And then photo 6 here, these oncoids are a little bit more complex because they are not necessarily reminiscent to what you would think of an oncoid being rounded or elongated, but they are coated grains. You have these incrustations, mostly composed form of Gyrvanilla, some tubiphytes, and some micritic patches that may have been formed via microbial activity. So as I mentioned, the oncoids are present throughout the entire formation, and this is very relevant. In order for us to come up with a depositional model, I divided my macrophages 8, which is the one that contains the most oncoids, into 4 submicrophages. So here we have macrophages 8A, which is a more proximal macrophages. It will be located somewhere in the inner ramp, at and below for weather wave base. And this contains encrusting forams, oncoids, and echinoderms. Then we have a little bit more distal macrophages here with more dasyclads, some fustulinids, so we're starting to get a more open marine conditions, and of course, oncoids. Then a bit more distal here, we have our oncoid, fustulinid, and calcareous algae. So here we lose the dasyclad algae, which typically in the Lower Permian, in Lower Permian successions, are interpreted as the shallowest kind of algae really that you can have. We are still within the photic zone, of course, and well above the thermocline, but this is a bit more distal and definitely open marine. And then here, our last macrophages that contains abundant oncoids is 8D, and this is definitely deeper water, open marine, contains fustulinids, echinoderms. I lose most of my algae. So in other words, if we have a ramp, something like this, we would have oncoids associated with different kind of organisms depending on where they were forming. There's other macrophages, of course, that I don't want to get into this because I don't have much time. But what is key here is that the oncoids if you were to swim in this kind of little ocean here in Austria, you will be seeing oncoids everywhere in the bottom. So what is interesting about the oncoids in the early Permian is that there's not only a single little occurrence, they are present globally. And what is interesting is, for example, here, this is our study in this Verdra Basin, which is located in Arctic Canada. During the early Permian, this Verdra Basin was located at about 25 to 30 degrees north along the northwest margin of Pangaea. And it was the shelves of this basin were a prolific photosol carbonate system. And what is interesting is that during the second half of the Astelian, and this is what we see here, we had what Bosham and Henderson call the oncoid interval. And it doesn't necessarily mean that there were no oncoids below and above this interval, but the oncoids were particularly abundant in this interval. And so what we see here is that this interval ends just below the Astelian-Sakmarian boundary, just below this sequence boundary over here in the Verdra Basin. Another important location is China, and this might be even more important for different reasons. First, here we have our graph showing different formations of Lower Permian age. The age of these actually are outdated, but what is key here is that the mapping and the Chuanshan formations, which are Astelian in age, are rich in oncoids. And so essentially the whole south of China, or the platform, the Yangtze platform, was covered in oncoids. And for us to understand a little bit what this means, if we look at this reconstruction here, South China would be somewhere here. And if we look at this reconstruction of the south of China, so if you can think of the modern China and you divided that by 2, you look at the south or southeast area, this is what we have in this photo, in this diagram. So the blue part here, the light blue part, this is the Yangtze platform. And this Yangtze platform was made of the mapping and the Chuanshan formation. So they're laterally equivalent. So if you picture the entire, almost the entire southeast or south of China being covered by oncoids, literally covered by oncoids, you can think readily that this had to have some kind of implication in terms of carbon sequestration from the atmosphere. So what is also important here in China is that the oncoids, or these oncolites, okay, thank you, also finished at the Astelian-Sakmarian boundary. Again, these ages are outdated. So what we are suggesting in this in this project is that the low CO2 concentrations during the Astelian were kept low, in part thanks to the proliferation of these oncolites, these oncoid beds in places such as China, this Verdra Basin, and other locations that don't have much time, but they also have abundance oncoids, thrombolites, stromatolites in the US and other locations during the early Permian, in the Astelian in particular. So these organisms kept CO2 concentrations low. The Astelian-Sakmarian boundary, when these huge oncolite beds disappeared, this major carbon sink disappeared, and that facilitated the rise of CO2 again. And then this is when our Zottachkopf Formation it's time to shine, because then these oncoids in the Zottachkopf benefited from this rise in CO2 concentrations, and they bloomed. So during the Sakmarian, because the Sakmarian is a transitional stage, there were not many locations where oncoids could form. And so in the only locations where they actually were able to grow, they did, because they had this increase in CO2 concentrations during the late Paleozoic, or after the late Paleozoic ice age. So that's the hypothesis anyway. And that's it. Thank you so much.
Thanks very much, Daniel. Have we opened up the chat room for questions? Yes. So you talked about cycles quite a lot at the beginning. How did you define the cycles in your field area?
So in the Zottachkopf Formation, they're actually quite easy because they're also, you can see them in the field. I wish I had photos of the Zottachkopf Formation, but you can see your sequence, they're bounded by sequence boundaries. And so what you see also in thin section is the repetition of at least more than 3 microfacies, right? So you begin with fairly deep water microfacies at the base of the section, going all the way to, in this case, ooids, to dasyclad microfacies, and then right on top of that, you have suddenly very deep microfacies again. It depends on where you are, so you may have some continental deposits as well, like in Kansas or Southwest US. There's a more classic cyclothems. But in this particular area, it was very good, healthy photozoan carbonates that made out these cyclothems.
Cool, thank you. And what do the sequence boundaries look like?
It depends. Sometimes they're very easy to miss. Some other times you may have something like microcodium or some karstic feature, so you understand that there was overall exposure. Sometimes it is definitely tricky and to identify the boundaries of these cycles. And when reading some literature, it's not impossible to find a different understanding. So where one cyclothem ends and where it begins. So yeah.
Yeah, no, I've had my own struggles in identifying cycles, so that's why I'm curious. Just to remind everyone that the chat is open for questions. I can't see that we've received any yet. Stephen, can you see any, or do you have any questions?
Sorry, I'm just dealing with a couple of issues here, but I don't see any at the moment, but the chat is open. There's one from Fiona.
Oh, Fiona's got one for us. Said, really interesting, thanks. Why were oncoids so abundant in the early Permian? And what do you think drove the reduction in oncolites in Arctic Canada and China?
So what I think that killed the oncoids in these 2 particular locations was actually a subarachnoid exposure event. So the ends of the Ascelian, or in the latest Ascelian carbonate package in this vertebral basin in the south of China, we have a major unconformity as well. Not as big as the Pennsylvanian Permian one, but we also have one. And then on top of that, we have the major transgression in the earliest Sackmerian, which was related to the deglaciation of the LPAA. So in my opinion, that essentially prevented these oncoids from keep forming. Otherwise, they would have been pretty happy in that location. So essentially for us to have abundant oncoids in a location, you need to have food for them. So what you need to have CO2, fairly low nutrients. You don't want to kill your system with too many nutrients. But you also have to have a good ramp where you have these guys moving around and all of this. So yeah, I think the fact that it was exposed and then we have this major transgression that kind of prevented these oncoids from keep forming in these 2 areas.
Brilliant, thank you. We have another one from Gene Ranke. He says, if you have waning glaciation, amplitudes of sea level change should decrease. What is the microfacies strat evidence for such a decrease?
Within one single cycle?
I'm not sure. Yes.
So in the early Permian, just in the current glaciation, the understanding is that sea level fluctuated by 120 meters. So it depends on the tectonic setting, of course, and the area where you're looking at. So in this vertebral basin, what I was seeing was going from cool water carbonates below the thermocline. So you can picture like 100 meters or more, all the way to subarachnoid exposure with microcodium and this very clean grainstone sometimes with ooids and bioclasts. So it really depends on where you are. Sometimes you can have just a mat mound that is then exposed. So yeah, it depends.
Thank you. Thank you for the detailed answer. Still have a few more coming in. I think we have a little bit more time. So we have one from Ismail. He says, thank you for your nice talk. What about sedimentary records of maximum flooding surfaces in your sections?
Yes, so that I have it in the Zottachkopf. So what is interesting about this area of this ramp is that the transgression here of the late Paleozoic Ice Age, you do have deeper water microfacies, but it's not a tremendous shift in phases like I see in China, for example, or Arctic Canada. I think this was also key for these oncoids to survive this transgression, because again, I do have oncoids on the MFS, or where I put the MFS. So again, because you're looking at very spatially far away locations, each of them have their own identity in terms of phases and evolution. And in this case, the ram never really got that deep. So what I see really on the MFS is kind of like echinoderms, bryozoans, in wax zones and pack zones. But it would still be above the thermocline. So we never cross below the thermocline in this particular location, which we do in this vertebrate basin, for example. But this location, because it was at the equator, very close, the thermocline is reasonable to believe that it was fairly deep, or deeper than at 25 or 30 degrees north, which is where this vertebrate basin was located.
Okay, great, thank you. I think that might be all the time we have for questions, though there are still some in the chat. So hopefully we can answer those in discussion later. We're having some, oh, he's back. Stephen's back, but I thought you were having some technical issues.
No, I was dealing with a few little ones from other people, but we're sorted out now. So,
okay.
Yeah, everything's back. So thanks very much, Daniel. Terrific stuff. I was listening. Next, we're going to Anna Claussen, and Anna's going to be talking to us all about Bryozoan-rich stromatolites from the Silurian. So, Anna, are you there? Yes, here's Anna. So if you could share, hello, could you share your screen, please?
Hello, everybody, and welcome to my talk. And also many thanks that I can have the talk here in the Carbonate forum. Today I will give a presentation with the title Bryozoan-rich stromatolites, or so-called bryostromatolites, from the Silurian of Gotland and their relation to climate-related perturbations of the carbon cycle. In older textbook, it was said that the Silurian was a common friendly period with extensive reef growth and without climatic perturbations. But researchers could show 20 years ago that this was wrong, and they did stable carbon and oxygen isotope measurements on brachiopods over the Silurian and could show that there were strong isotopic shifts. And one of them, it's this one, it's called Low excursion, and it's the strongest for the whole Phanerozoic. And it's still under debate what was driving these isotopic excursions. My supervisor was involved in this research, and yeah, he did a lot of his research on Gotland, and there they found a really strange reef type, which can be seen here, and it has a cauliflower surface structure. It's really hard to spot also in the field, and these reefs were forming small patch reefs, which are distributed over the outcrop. And in thin sections, they are mainly composed of bryozoans and microbes. This is why they are called bryostromatolite. And this is a facies which is completely new, although Gotland is really well studied. So I'm doing my PhD project at the moment, and there I first want to characterize bryostromatolite as a facies type. And also I want to study Silurian bryozoans, because bryozoans are really underrepresented in research. So I also want to show that that can be also really interesting. So the aims are: how do bryozoans react to the Silurian geochemical events, and what is the eventic response? What caused the formation of the special reef type bryostromatolite? Do bryozoans also show an enhanced number of malformations related to stable carbon isotopes excursions? So this is something that could be shown for aquiters and Pitinozoans. So I also wanted to find out if also bryozoans do the same. And I also wanted to have a look if single species differ in shallow compared to deeper water settings, in, for example, the size, growth form, or wall thickness. How different are coeval communities between the Paleocontinent, Laurentia, and Baltica, also to see if the communities are mixed and if there is genetic exchange between them. And why was the bryozoan diversification so suppressed in the Silurian in comparison with Ordovician and Devonian? Because the diversity of Silurian bryozoans is declining compared to Ordovician and Devonian and other marine biotas. It's something like this cannot be observed. So the question is this a signal or bias? So, and I want to focus on the first question here in this talk. So this is how bryostromatolites look in the field. And here you can see a clean sample from such a bryostromatolite, and you see this cauliflower surface structure, how it's developed. So this is the stratigraphy of Gotland. Gotland is an island which is located in the middle of the Baltic Sea and belonging to Sweden. And on Gotland, the complete succession from the end of Tertian to the end of mid-Permian is exposed. And on Gotland, I sampled in 77 localities distributed all over the islands, and 10 of them have bryostromatolites. So they are quite abundant. Our hypotheses are that bryostromatolites occur solely at times of elevated stable carbon isotopes, and that bryostromatolites, if this is true, are a time-specific facies, and the formation is restricted to extreme and extraordinary environmental conditions. Time-specific facies means that this is a facies that is only occurring in a narrow time interval, which is linked to an event. So to test the hypothesis, we first measured the stable carbon isotope ratios of micrite and bacteropods from the bryostromatolites. So the bryostromatolites are here, and they have the following distribution. 3 of them were found at the Iván isotope excursion, while 7 are found at the Lower isotope excursion, where we found no bryostromatolite in the area between this. So to see if this is a real distribution or artificial, we also compared this with literature examples and compared this with Ordovician and Silurian examples. So here again are bryostromatolites, and these are literature examples. And the reefs there, they're having a really similar distribution, except from the one in the Basin-scale formation. And this reef is the earliest known Metazoan reef, and this is why we can assume that this is an exception. But we can say that more or less the hypothesis seems to be correct. So to test the other hypothesis, we compared the bryostromatolites to find comparable features. And this is a thin section from one bryostromatolite from Bangor. And to highlight the components, I added some color. And you can see that the bryozoans, which are displayed in red, and the microbes, which are displayed in gray, they are dominating the reef. Common Silurian reefs are formed by rubose corals, carbonates, stromatolites, or maybe algae, but here the composition is totally different. And you can see that in these downwards facing parts, how the cauliflower surface is developed. The reef was able to perform the growth in every direction, but also downwards. Okay, we have also observed other interesting features. So this is a crust-like structure composed by spheroids or globular shapes. Maybe you can see this here. So they are really globular, and we sometimes have also rosettes like here. And this structure is called paleomicropodium, and this is something that is used in the literature for indicating extreme environments. We have in fenestrial cavities cross-shaped crystals, and they are called hemibipyramidal twins. and they are formed by gypsum, but now the gypsum is recrystallized during the diagenesis and replaced by calcite. But the presence of gypsum indicate that we had subaerial uplift of probably also aridity. We have a high abundance of phosphatic remains in the bryostromatolites. So we have 3 different species of phosphatic brachiopods that can be observed, and 4 bryozoan species that are precipitating phosphate. Like in this example, this is Fabrizitella, and here the Fabrizitella is forming pearls. But it's not just one pearl, it's really a massive amount of pearls. And phosphate is nowadays a really limited factor or nutrient, but here it was so enriched that the organisms had to get rid of it. Yeah, but it's hard to assume how the water was in Paleozoic times. But we can assume that we had some kind of a plethora in the bryostromatolites. So, yeah, possible sources are dust, weathering, and precipitation, but we cannot say what exactly drove this here. So our conclusion are, yeah, the bryostromatolites are completely different compared to other Silurian reefs. We have paleomicrochorium, which is indicating extreme environments. We have pseudomorphs after gypsum, which indicates subaerial exposure and aridity. And we have phosphatic remains, which are indicating a plethora of phosphate. So we can assume that we have had a time-specific phase because the reef was totally different compared to the other reefs. And to highlight the significance, our bryostromatolites are extraordinary and so far undescribed for Gotland, so they are totally new.
2 minutes.
And they were found under extreme conditions and assumingly with increased phosphate, something like phosphate plethora is not before discussed in combination with stable carbon isotope excursion. And the bryostromatolites occur only at times of elevated stable carbon isotopes. So, and by this we get new insights in the fully understood isotopic shifts of the Silurian. And if you are more interested in the work, we have recently published a manuscript. This you can look up online. So thank you for attention. And if you have questions, please ask them. Thank you.
Thank you very much, Anna. Why don't you go back one slide and really advertise that paper you've just published? There you go, so everybody can get a good look at the title. I'm learning English. Right, okay, brilliant stuff, absolutely brilliant stuff. Can I, with the questions open, please, people do start typing your questions. I will speak slowly to give you a chance. While you're doing so, just one quick question from me, more on nomenclature really. When do you stop calling something a bryozoan stromatolite and start calling it a bryozoan reef? What's the difference there? Because you did use both terms, I noticed.
Yes, so bryozoan reef. So when you just look at the bryozoans, we had first to make a definition with what is a bryostromatolite to understand this better, also to understand what we have there in the field. So it is really important that we have a lot of stromatolitic or at least microbial input in this reef. So it is not just that we have some microbial crust. We have porous stromates, we have spongious stromates, so it's really full of this. And bryozoan reef, when the microbes are not reported for them, then we would assume that this is not a bryostromatolite, but this is something hard because taxonomists would maybe only report the bryozoans. There's also the problem with the literature. So, it would be good because we compared them with artificial examples to have a look there in the reefs if this really is a bryostomatolite or if this is just an assumption that it could be.
Yeah. Okay. No, obviously, it's my bad. It's my bad. I obviously need to read that paper, so I'll get on with that. Our first of our question from the audience is from Jean Ranke. Interesting talk. Could the phosphate alternatively be due to upwelling? That would also impact the delta 13C.
Sorry, can you repeat this?
Sorry. He says, interesting talk. Could the phosphate alternatively be due to upwelling? That would also impact the delta 13C.
Yeah, this is something. It's a good question because the phosphate content is increased when we have upwelling. The problem is that we're up here, and yeah, we are on a shallow platform, so something like upwelling cannot happen here, and this is a really big problem. Yeah, so we cannot expand when we have the phosphate increased. So it must be something different, like dust or weathering, or yeah.
Okay, that's great. Please, people, do ask your question. I have a feeling everybody's the after lunch sleep has started kicking in. So please, people, wake up a little bit. Let's see those questions coming in. Lucy,
sorry,
did you have one?
Oh, just a quick one. I really loved the image you showed with your where you'd highlighted the different fabrics and elements of the bryostromatolite with different colors. Did you do that all by hand, or did you use some kind of image recognition?
Oh, I was doing this all by hand. It was a lot of work.
Like it. I wonder if something, we could in the carbonate world develop something to recognize those features, because it looked really cool. But I imagine very time consuming.
Yeah, it was. So this picture takes, I think, 1 day. So
yeah, well, it looked great.
Thank you.
I think we've all been there on those long-term pictures. If I may, I'll just take the advantage of nobody else asking a question yet. The gypsum, you showed the gypsum. What's the timing of that gypsum? When's that gypsum appearing?
Yeah, our assumption is that okay. First of all, we assume that the reef was formed directly at the sea surface. So this is indicated by paleomicrocodium, which is assumed to be terrestrial, and it's also there. And it is, after it is formed, uplifted somehow. But yeah, we have cavities with paleomicrocodium in, but also cavities with the gypsum. And so we assume that this happens shortly after. But yeah, we also assume that it's during diagenesis also gone really fast.
Okay, well, thank you. Any more questions? We've got, I've had one sent direct to me. Please, just a little gentle reminder, everybody, please do send your questions to everyone, but I'll read this one. This is from France. What have we got? Based on the hand specimen in the thin section that you showed, there aren't many laminae present. Stromatolites are characterized by laminae. Why the biostromatolite name? Maybe biomicrobialite, or would be more appropriate.
Yeah, okay.
And then, sorry, then an apology for the semantics.
So I think this is more general question also for stromatolites. So not for also bryostromatolites. So we have some thin section where we have a lot of porostromites. It's a bit something that I haven't shown you is that we have time succession inside that we have had. So maybe I can show this. Yeah, we have made a reconstruction of the reef. So we have first a phase where we had corals, so typical Silurian reef, stromatoporites, and afterwards the corals were dead and other organisms and covered by porostromites. So this is the first, let's say second phase. And after some time, also this is covered by these bryozoan crusts together with spongiostromates and forming cryptic habitats. So it's really complicated. Yeah.
Okay, well, in fact, we're still stuck on this question of semantics for our last. This is going to be our last question. I've just received time being called, but thank you. This is from Tanya Unger. Thank you, Anna. Great talk. I am not 100% into bryozoans systematics, but can you determine a change in species in your bryostromatolite?
A change in species, so
Yeah.
from the bryozoan species.
Sorry, yes, they've added then compared to the bryozoans in the surrounding fascies.
Oh, this is something where I'm at the moment working on exactly.
Okay.
So, but it seems like we have different species. So first, it's surprising we have a high diversity. So we have between 12 to 21 species, and yeah, some of them are really, really special. We have never seen them before. So yeah. But this is something I'm working on at the moment.
Terrific, terrific. We can carry on these discussions again in the discussion section after this session, and I'll hand you, thank you, Anna, thank you again, and I'll hand you back to Lucy.
Brilliant. Thank you very much again, Anna. That was great, something I'm quite unfamiliar with. So next we have a talk from Abiotic. Are you there, ready to take the stage? With the talk entitled Warming-induced Abiotic Carbonate Formation in the Mediterranean Water Column. I can see you there.
Should I share my screen?
I think you should be invited to share it.
Okay. No,
you should already have permissions, or I'll just check.
No problem. Okay, here we go. Okay, hi everybody. My name is Or. I'm a Marie Curie Research Fellow based currently out of the University of Malta. And I want to take us up from the water, from the seafloor, and go up into the water column for a bit. And I want to start with a little bit of something we all know. And that is that most of the low latitude surface ocean is supersaturated in respect to calcium carbonate. We usually annotate this supersaturation as omega, where omega is larger than 1, we're in supersaturation. But while we do have the supersaturation, we don't usually get to situations where calcium carbonate is being precipitated out of the water column. There are some kinetics involved with that, limiting that, but there are exceptions. I think one of the most spectacular exceptions to this is whiting events, which are occurrences where we have basically the water turning milky because there's a lot of fine-grained calcium carbonate precipitated in the water bodies. The reasons for these events are contested, and people have been arguing about this for many, many years, but we have them well documented in a few locations, the Bahamas, I think, where they've been studied the most, as well as the Persian/Arabian Gulf. There are also some possible indications for them in the Red Sea. But these are the exceptions. Most of the time, when we go to the open oceans, we don't see anything that is clearly carbonate being precipitated without biology from the water column. And then we need to also think about some of these kinetic aspects. And this plot has temperature in the x-axis and aragonite supersaturation in the y-axis, and the contours are showing the rate of aragonite precipitation. And this is based on this equation, which shows that the rate is a function of several components, including saturation, but the other two are temperature dependent, from Burton and Walter, 1987. But within this space, we also need to remember things that are going on right now. Primarily the fact that people have been dumping a lot of CO2 into the atmosphere the past 200 years. One effect of that, which affects the water chemistry as well as being a major problem for calcifiers, is acidification. Acidification brings the saturation state down, and that naturally would reduce the rate by a lot. But there is another force coming into because of that, and that is global warming. Well, warming, if you look at this equation again, would actually increase the rate. So essentially, at the end of the day, we're getting a vector sum of these two forcing when we look at modern oceans. But that doesn't necessarily mean that we're getting a net increase in the potential rate of aragonite precipitation, because I'm drawing here a warming of 4 degrees. That's a lot. We don't have that many places in the world that see that level of warming. Well, let me take you to the Eastern Mediterranean. The Mediterranean is a marginal marine basin, one of the largest in the world. And it's also very long, which gives us a very, very significant gradient. Water come into the Mediterranean from the Atlantic. They're 17, 20 degrees in summer. They're at 35, 36 PSU. By the time they cross all the Mediterranean and get to the Levant Basin, which is the easternmost basin, they're 30 degrees centigrade. They're 39 PSU. So they're a lot saltier and they're a lot warmer. And they're also very, very nice and clear. This is a picture taken out of Mount Carmel. You can, in the background, you can see how blue everything is. But over the last 40 years, for which we have instrumental records, we see that these waters have experienced a lot of warming, about 4 degrees worth of warming, so about 1 degree centigrade per decade, which is a ridiculous amount of warming. And as they warm, they also means that through their path, they evaporate more, so we also slowly see an increase in salinity. And because these waters are very, very oligotrophic. You can see here the primary production in the water column over a yearly cycle from a recent preprint that is out now. And you can see that even during winter with top production, we're talking about 3 milligrams of carbon per meter square per day. And in summer, we're talking about less than 0.5 milligram, which is possibly the lowest primary production in the ocean surface anywhere in the world. And we also see a very, very strong stratification. This is one of the reasons for this intense warming in the sea surface. So the bigger plot shows temperature over a year, with the contours being with the y-axis being depth and the contours being temperatures. And you can see this formation of this upper stratified water column with very, very high temperatures getting to 30, 31 degrees centigrade. So it's very warm, it's very stratified, it's very oligotrophic, it's very salty. And if we take the record further, we can take it down all the way back to the 50s with instrumental record for going forward. Because of this oligotrophy, alkalinity is very, very conservative. So net alkalinity has been going up and up and up since the 1950s. But because acidification is a thing, since we have a good reconstruction since the damming of Aswan in the 1960s, aragonite saturation has been going down. Which brings us back to this. So acidification brings saturation down, while at the same time global warming, and especially the stratification and the development of this very warm upper water mass takes us into higher precipitation rates. So net effect is that this basin has seen an increase of about 16% in the potential rate of aragonite deposition, aragonite precipitation rate. So we started to think about this, and we started to hypothesize that if we see such an increase in potential rates, and also with the stratification, we see a lot of CO2 degassing, maybe during these warm stratified events, and with this extremely high alkalinity, maybe we do get some precipitation of carbonate from the water column. And we were very lucky because there was a set of traps that was deployed during 2015 and 2016 within the stratified part of the water column a few kilometers offshore. And when I started analyzing this record, well, the first thing that popped out was when we did the XRF, and we saw the strontium calcium ratio shoots up in the summer, then goes down, and then shoots up again. Well, strontium can be a lot of things. So next thing you do XRD. And when you did the XRD, well, we didn't see that much for 2015, but the background was very high because of other things there. But in 2016, the levels went very high, and there was a significant flux of aragonite. Now, we know this aragonite is not coming from pteropods because, again, primary productivity is very, very low, and we have data from NATTOs showing there are no pteropods in the water column, and that also doesn't fit with the strontium. So the next thing is, oh, there are these vermetide reefs next to the coast. Maybe they're getting reworked during summer storms or something like that. So we looked at backscatter data from an ADCP that was next to the trap, and we see that in winter there's a lot of suspended material, but as these aragonite levels are coming up, it actually goes down. It's very flat during the summer. So we don't get a lot of suspended material coming in during that time. So it's not biology, it's not getting reworked. Our only other option is, well, this is probably this chemical signal we saw, and that's great. But next thing is, let's check this against saturation state.
2 minutes left.
And when we look at the saturation state over the year, we saw quite nicely that in 2016, the levels went up as the aragonite in the traps went up. So why not? We don't see anything in 2015. We think the issue is induction time. So during 2015, there were these cooling events during summer that broke off the stratification. And when you look at the induction time needed to start generating aragonite for the level of saturation in seawater, we're talking about an order of 2 months. The reason we don't see this in the sensors that was attached to the trap was probably because the warming starts higher and then creeps down with stratification. So it's probably started earlier, and that's why we're starting to get aragonite already a bit before. So what do we think is going on here? During winter, we got the trial material coming in. We got a mixing of the water column. Temperature is low. As we're getting into the summer, we developed the stratification. The upper water column developed a thermocline about 10 degrees warmer than the water above it. These waters are super saturated toward CO2. They degas, and in that space, that's probably where we're getting this aragonite precipitation. It also shoots up the omega locally. It goes above 4. Whereas during the rest of the year, it's not. During the winter, it's not. So to summarize, abiotic carbonate precipitations occur in the upper water body of the warm, salty, and highly alkaline, in oceanic terms, southeastern Mediterranean, in years during which stable stratification is established in the summer. What does this mean? Well, for the modern ocean, we think this means that anaerobic precipitation events are probably more prevalent than we previously thought, and they don't necessarily have these features in the upper water column. Maybe they're occurring deeper on the thermocline. So maybe let's call them crypto-whiting. And as the oceans become warmer and more stratified, we should expect more of these. And for the past ocean, well, specifically for empiric seas, which are very shallow, very warm, very supersaturated, it is most likely that a good chunk of the aragonite we're seeing there has an abiotic component, which goes back to arguments made by Peter Swart that we need to think again about things like what does bulk delta C13 from platform tops actually mean. And with that, I'll thank you for listening and open the stage for questioning.
Thank you very much. Yes, the chat's now open for questions. Can you just flip back to your conclusion slide so we can see the main, or the slide just before this one, just so we can see the main points. Just the next one after summary, sorry.
So this one.
I think it's just moving slowly. Yeah.
Okay.
So I was just going to ask, so you say this process may have had a much more prominent feedback effect on climate regulation. Could you just maybe expand on that a little bit? I'm quite interested in what you mean by that.
Okay, so there are a few things that are going on here. Well, and the first one is that calcification actually acidifies the water, because we're taking most of the carbon, the dissolved inorganic carbon is in bicarbonate, so we need to take that into carbonate, which discharge a proton into the water, resulting in a net decrease in pH. So that decreases the water mass capability to store CO2, and it also may have a limiting effect on calcification itself. So it's two factors playing one against the other. The other issue is when you do this, there's actually a net CO2 release. So if you warm these epicontinental seaways a lot and you start this process, you might actually release more CO2, which is also true in general when you warm, if you have a warm stratified upper water mass that's detached from the water below it and get to a certain temperature, for specific values of the atmospheric PCO2, it will degas, which is, for example, what we're starting to see today in some marginal basins.
Thank you. Thank you for the detailed answer there. We've started to get some questions in the chat now. So firstly, from Megan Smith, many fish secrete calcium carbonate in their gut as part of the osmoregulation process. Have many fish secreted calcium carbonate in their gut as part of the osmoregulation process? Have you considered this source?
Yes, we have considered that, and we looked at the catch data and fish radar information that the Oceanographic Institute has. And during the summer, this is the period where we see the least activity by fish to actually try to go below the stratified layer, because that's where the food is. So in the upper water mass, you don't get a lot of fish activity.
Mm.
So the odds that what we're getting is coming from fish activity is very low. And we looked also at other species, and everything just goes down during the summer, especially in the stratified water column, because it's really, really warm. Not a lot of things want to be up there. And so that actually limits the results a lot. Also, we looked at the morphology of these crystals, and they don't look like, and they don't form the aggregates and cluster that we expect from other biological activities.
Interesting. So yeah, you've definitely looked into that one then. One from Chelsea. Thank you for the interesting talk. I was wondering if you've measured other proxies, calcium, alkaline, etc., over an annual period to directly calculate how saturation state varies over time in the upper water column.
So we did an overdefined time series over a year, measuring pH, alkalinity, and DIC. We still haven't done calcium and magnesium. That is planned for next year, but that's something we are thinking about. And we do see a cycle through the year, and we do see some changes in alkalinity. We're still calculating this because it's a bit tricky to deconvolve some of the things going on. But yeah, we do see an annual cycle. The outgassing of CO2 during the summer is sort of oversaturating everything. So it's kind of hard. So it's kind of hard to deconvolve exactly what we're getting. But we have calculated, and we get that a certain fraction of the CO2 we're getting out does exceed what we expect from thermodynamics. And we think that component fits more or less to what we expect to get from the calcification.
Okay.
But we're still working on that one.
Yeah, it sounds like it's on the cards for next year then. Next question from Kathy Hollis says, thank you, or that was a great talk. So just to be sure I understand, even if a short period of cooling during 2015 was enough to prevent abiotic aragonite precipitation?
That's what we're thinking, although, again, we don't have enough experimental data on induction time, especially in lower level of supersaturation. If anybody wants to take a student with me and do those experiments, that would be great. But the problem is they're going to be very, very long. It takes a couple of weeks to get to induction time at this low level of supersaturation. So if we go with what happens when you break induction time with other things which are faster, it seems that, yeah, that basically resets the timer. And we need to wait again until we get enough days of continuous supersaturation in relevant depth to get to initiate this reaction. Once it initiates, it seems to be going quite nicely. But if we break it, it seems to be either very suppressed or not at all. And again, we need more experimental data.
Yeah, always the way. I think we have time for one more question. From Ismail. So thank you for the nice talk. What about rain precipitation, sunshine duration, and wind evaporation rate within the studied interval in the Mediterranean? Were they considered?
There is almost no rain during summer in the Mediterranean, so that's off. There are different, so wind and evaporation is something that's going on, but the change in salinity we observe is very, very small overall. So we don't think it's just pure evaporation unless there's some threshold which we don't understand very well, which is always an option with carbonates. So it's probably not that. Does wind and aerosolization play into this in some way? Maybe. I wouldn't discount it. It's but we don't have the level of nuance in the sediment track record to specifically correlate with individual wind events. That would be great to have longer time, longer and higher resolution trap series. I'm trying to get that, but those are very, very expensive and take a lot of time to set up.
Okay, brilliant. Thank you very much. I think that's all we've got time for your talk there. Or Stephen, over to you.
Thank you very much, Lucy, and thank you all. Terrific presentation again. Right, okay, next we have Iván Díaz-García. In fact, we don't have Iván, unfortunately. Ivan's not been feeling too well. So the scientific committee have had a chat, and under the exceptional circumstances, they've okayed for Ivan's supervisor, who is Oscar Marino Toma, to present on Ivan's behalf. So you can, oh, you've already done it. Share your screen and please go ahead. I'm afraid, Oscar, we're not actually hearing anything from you. Could you please make sure that your microphone is turned on? It doesn't appear to be turned on from what I can see. And your video as well. Okay, Oscar, can you please try and turn your video on?
Yeah, I'm trying to do it. Okay.
Okay,
Sorry
there we
for
go.
the delay.
Right. Okay, fine. So it's over to you.
Yeah, first of all, I would like to thank you for allowing me to do this presentation. And I'm going to summarize this fantastic work that Iván Díaz-García is making during his PhD, but unfortunately, he is not available to present this talk today. Iván is working in a very enigmatic carbonate succession of mid-Carboniferous age that is present in the north of Spain and that was accumulated in a very broad foreland basin during the assemblage of Pangea. It's an enigmatic succession because it's mostly composed by carbonate mud that is now recrystallized into microsporite and sporite that has common pelagic microfossils like radiolarian biomolds and other microfossils like globochaetetes, pelagic ostracods. And one of the remarkable features of this succession is the common presence of calcite pseudomorphs after gypsum intrasediment evaporites. So one of the difficulties in his PhD is to find comparable examples in the literature. Nowadays, we can find intrasediment gypsum evaporites in coastal marine successions and in continental successions. But it's rare and it's not very common to find intrasediment gypsum deposits in old deep marine evaporitic basins that usually are characterized by the presence of selenite gypsum deposits. The sample I'm going to show you was developed in a subtropical environment in a very broad basin that was located more or less at 20 degrees south. Now we have some examples of the sedimentary successions accumulated in that very broad foreland basins in the north of Spain, in the northwest of Spain, in the so-called Cantabria Mountains, and in the Pyrenees in Spain, and also in the south of France. And we know that southward in the same sedimentary basin, there were evaporite deposition at the same time interval. There are some evaporites in Algeria, in the Tindouf area, and also in areas close to Colombes-Becar. This is more or less a transect that shows a correlation of the stratigraphic successions that were locked in these successions from the north of Spain in the Cantabria Mountains to the Pyrenees. This succession is reconstructing the thickness of this Carboniferous carbonate pelagic succession that is thicker in the areas closer towards the foreland, towards the foredip of the foreland basin, and gets thinner along a very long area towards the foreland of the basin. This transect is more or less 300 kilometers in width, previous to Basin-scale deformation. 500 kilometers away to the east, we found the same stratigraphic intervals in the Pyrenees. This succession that can be up to 400 meters in thickness and that interfingers with turbidites in the foredip areas of the basin is only 60 meters in thickness in the foreland and contains a very continuous, with a very remarkable uniformity in thickness interval that has this gypsum, intrasediment gypsum evaporites. I'm going to show a correlation along this interval in the next slides that shows more or less the thickness of this stratigraphic package with the evaporites, in which we observe an increasing abundance of evaporites from the base of the interval towards the top. The abundance of evaporites is so high in the uppermost part of this interval that is locally higher than 70%, 60%, that the interval used to be vitiated, and it's very uniform in lithophases. And below this interval and above, we have pelagic carbonate successions. I will show you an image in the following slides. And the evaporite deposits consist mostly of intrasediment gypsum evaporites precipitated within carbonate mats containing radiolarian biomolds. There are some microbial-like lithophases with some intrasediment gypsum evaporites. And there are also some gypsum cumulates. And towards the foredip of the basin, we have observed also detrital gypsum in turbidites. This is the appearance of the pelagic calciumostones that comprise the evaporites. They are mostly mudstones and wackestone recrystallized into sphaerites and microsphaerites with common radiolaria. This is Aperitis radiolarian. And near the base of the evaporitic interval, we used to find dispersed evaporites formed by calcite pseudomorphs after gypsum crystals that occur dispersed within the laminated calciumostones. This is an image from the northwest of Spain. This is another image of a sample from the Pyrenees. Crystal size, crystal geometries are very uniform across the entire basin trunk set. Upwards, as the abundance of intrasediment gypsum evaporites increases, the size of the crystals decrease, and some concentrations of evaporites define a laminated microphases, but also having the radiolarian biomolds. Locally, only locally, we have observed some gypsum cumulates interfingered within the intrasediment gypsum deposits. And in some locations of the basin, usually towards the foreland, but locally also in an area closer towards the foredeep, we have observed microbial-like lithophases, very rich in radiolarian biomolds that has intrasediment gypsum crystals, now replaced by calcite, and also bottom growth like gypsum crystals associated with filaments of potential microbial origin. Towards the foredeep, in this area, we have turbidites interfingering with the pelagic calciumostones of this succession, in which we can find, in addition to detrital terrigenous grains like quartz, tourmaline, and other lithoclasts, we observe calcite pseudomorphs after gypsum with lenticular and prismatic geometries. This bed in these samples is interfingering within intrasediment gypsum evaporite deposits. In some locations, only in 3 of the more than 40 outcrops studied in this study, we have observed that on top of the evaporite interval, there is a remarkable subaerial exposure that resiates the intrasediment gypsum deposits, forming a nodular breccia in which we can find usually dissolved intrasediment gypsum crystals like this one that is filled at, or that show geopitals infills in which we can find root traces that can be seen here in cathodoluminescence. So one of the difficulties in this case study is to find a model that can explain on one side the accumulation of intrasediment gypsum crystals in relatively deep subtidal environments located below the fire weather wave base, and the presence also in the same deposits of radiolarian biomolds, another pelagic bioclast that suggests that at least during certain periods of time, the uppermost waters of the basin had normal salinities. So the other difficulty is the fact that these intrasediment gypsum deposits had not been commonly described in ancient samples of marine evaporatic basins. At least the deep basins with evaporated.
You have 2 minutes.
Yeah. At least the samples like the Mediterranean basin during the Messinian salinity crisis is characterized mostly by selenitic deposits. So we come out with this model. It's a model in which we explain how the pelagic calciumostones with the radiolarians could be formed in the basin, and how the intrasediment gypsum could have precipitated within those pelagic sediments. This model shows 2 stages that alternated during the deposition of the evaporites. A wet stage in which the superficial waters were had lower salinities and could be inhabited by radiolarians and other pelagic microfossils. And a deep water brine may be subsaturated, may be saturated in gypsum, but during this interval, a gypsum precipitation was not possible in the bottom of the basin. This wet stage alternated with dry stages in which the evaporation increased the salinity of the superficial waters until they reached the same salinity that the deep water brings. At that time, a mixing of the two layers of waters occurred, and increase in evaporation allowed an increase in salinity of the brine until the saturation of gypsum could be reached. At that stage, the deposition and precipitation of the gypsum in the sediment started. Following this evaporative deposition stage or interval, there was a significant sea level fall that allowed the development of subaerial exposure in some areas of the basin. This subaerial exposure can be timed using the available biostratigraphic information, and is more or less can be correlated with the mid-Carboniferous boundary, in which a significant sea level fall is recorded in many basins and many shallow water carbonate successions around the world. This is the case of the Donetsk Basin, in which a significant marine offlap is recorded. This is the mid-continent succession. And this sea level fall has been estimated by different authors to reach up to 140 meters. This is the sea level fall that might have followed the evaporated position, and that maybe restricted the basin, allowing the deposition of the evaporites studied by Iván. So thank you very much for your attention. And now I'm ready for your questions.
Okay, everybody, thank you, Oscar. Everybody, please do type your questions. Please don't forget to type them and send them to everyone. We're still getting questions just coming to me. So please do send them to everybody. We all want to see those questions. While people are typing, I just want to, sorry, I'm doing a few things here, a few balls in the air. One quick question for you, Oscar, while we're waiting for the first questions to come through on the tweet. Can we go back a few, just 3 or 4 slides to your model, just showing the basin with the evaporation, where you actually put the percentage evaporates? Yes. Yeah, that's the one, that's the baby. I was a little confused, maybe I missed something earlier, but you're at the bottom of that diagram, the 2 lower diagrams, it appears to be showing what look like turbidites moving up onto the slope. Are these, could you explain that a little bit, what the process is that's happening there?
Yeah, I'm aware that this is probably a drawing that is not correct. Obviously, the turbidites probably were accumulated downslope, but in the diagram that we did, erroneously we draw the turbidites going upwards. We are aware that evaporites, that turbidites were interfingering with the studied pelagic successions, but probably this part of the cartoon is not correct. So we should have drawn this downslope.
Okay, so they are moving down. So I was just wondering if they had momentum and they were just hitting toe of slope or something from another source. Okay, well, we haven't got any other questions at the moment. Lucy, do you have any, or can I keep
right now?
All right, I did have those turbidites, the actual turbidites themselves. Are those gypsum turbidites? So those clastic gypsum, or are they a mixture of clastic gypsum and carbonate? What's the actual composition of the turbidites?
Well, we have observed several samples. There are at least 2 stratigraphic sections in which we have found these turbidites, and usually we have some terrigenous material that was sourced from the orogen, coming from the active margin of the sedimentary basin. And we also found some carbonate material. In addition to that, we observed in the thin sections a large number of prismatic and lenticular molds, now replaced by blocky calcite, that we interpret that might correspond to detrital gypsum. And for us, we interpret that this gypsum probably was sourced from the orogen-ward margin of the Foreland Basin.
Okay, thank you very much. Please, people. Pete Burgess. Oh, they're coming through thick and fast now. And I had another really good question. I'll have to ask you in the discussion. Pete Burgess, interesting talk. Sorry if I'm looking at an angle. Interesting talk. Thank you. 300 kilometers, quite wide for a foreland basin, at the high end of the range for that type of basin globally. Any thoughts on why the basin is so wide?
Well, this is an interesting question that I'm not sure to be able to answer. This is something that is interesting of the Variscan Foreland Basin. It's a very, very broad foreland basin. Now we have to reconstruct the width of the basin, just restoring the tectonic deformation, because the successions in filling the basin has been highly deformed during the Variscan orogeny. But we know that the basin was very wide. Even the outcrops we are studying just represent a portion of the foreland basin. The basin could have been so wide maybe because the Gondwana lithosphere, that is the substrate of the foreland basin in this area was maybe very rigid, and as such the tectonic load imposed by the Variscan orogen were able to draw a huge area of the Gondwana's margin. Maybe that can be one of the possibilities.
Excellent, thank you. A question from Fiona. How does the physics of turbidite flow change in response to an increase in water density during periods when salinity is higher and evaporites form?
Oof, this is a difficult question. I'm not sure to be able to reply to that question. We have observed in the four deep successions the presence of turbidites that usually don't develop the typical Bowman sequence, despite the green side is fine or should be have this Bowman sequence. Usually some of the turbidites has a sharp top, and the TE is the transition from the TC to the TT interval is very sharp.
Okay.
I cannot really provide a very clear answer to that question, but apparently there was something in the density of the waters that make these turbidites a little bit different to what could be expected in normal marine waters.
Thank you very much, Oscar. I do see that we do have some more questions, but we do actually need to move on because we're a little bit behind time at the moment. So thank you again. Before I welcome Fiona back, and where is she? There she is. Before I welcome Fiona back, I just want to thank my co-host. So thank you very much, Lucy. And thank you to all presenters in this section. It's been a cracking session. I'm hoping to carry on the discussions in that discussion session that Fiona's about to announce. Thanks, Fiona.
Yeah.